Did you know that there are over 43 different schemes for typing blood into different groups? Fascinating, isn't it? These systems are based on the presence of various glycoproteins, lipoproteins, carbohydrates, and other proteins. However, not all these systems are equally significant or studied. Today, we'll be exploring some of the most common blood group systems recognized for their clinical relevance. These include the well-known ABO and RH systems, the MNSs system, the P system, the Glycophorin system, and the KEL system. Each of these systems divides human blood into different types based on specific antigens and their corresponding antibodies. Understanding these blood groups is crucial not only for safe blood transfusions, but also for understanding certain medical conditions and genetic traits. So, let's dive deeper into these systems and understand their significance. The ABO blood groups system, the oldest and most important of them all. It's a cornerstone of our understanding of blood compatibility. This system categorizes blood into 4 primary types: A, B, O, and AB. Now you might wonder what these letters stand for. They denote the type of antigens, proteins that trigger an immune response, present on the surface of your red blood cells. Type A blood has A antigens, type B has B antigens, AB has both, and O, interestingly enough, has none. These antigens are like ID tags that help your body recognize its own cells, but they also play a pivotal role in blood transfusions. You see, if you receive blood from a type that's incompatible with your own, your immune system will wage war, leading to a potentially dangerous reaction. That's the ABO system for you, a crucial determinant of blood compatibility. It's not just letters and symbols, but a lifeline in medical emergencies. Next up, we have the RH blood group system. This system is a vital part of our understanding of blood types, second only to the ABO system in terms of its importance. The RH or rhesus system classifies blood as either RH positive or RH negative. This distinction hinges on the presence or absence of a specific antigen, the RHD antigen, on the surface of red blood cells. But why is the RH system so crucial, you might ask? Well, it plays a fundamental role in blood transfusions and pregnancies. For instance, if an Rh-negative individual receives Rh-positive blood during a transfusion, it can lead to a harmful immune response. Similarly, during pregnancy, an Rh-negative mother carrying an Rh-positive fetus may develop antibodies against the fetus's blood, leading to potential complications. The RH system, a critical factor especially during blood transfusions and pregnancies. There's more to blood groups than just ABO and RH. Let's begin with the MNS blood group system, an early detection from 1927. This classification is based on the 5 key antigens M, N, S, S, and U. However, the system recognizes a total of 50 different antigens, encoded by the Glycophorin A and Glycophorin B genes on chromosome 4. Although antibodies against these antigens exist in our blood plasma, they're generally clinically insignificant. Next, we have the P blood group system, also discovered in 1927. This system segregates blood into different types based on the P-type carbohydrate antigens, controlled by the B3 GALT1 and the A4 GALT genes. Among the 5 antigens, P, P1, and PK are vital as they can cause transfusion reactions and erythroblastosis fetalis, a condition affecting fetuses and newborns. Moving on, let's explore the Glycophorin blood group system. Here, blood is classified based on the 19 Glycophorin antigens present in the red blood cell membrane. These antigens are encoded by variants of the BCM gene, and the system operates on 2 codominant alleles, LUA and LUB. Although this antigen and corresponding antibody system are rarely associated with transfusion reactions and erythroblastosis fetalis, their presence is crucial to note. Finally, we have the KEL blood group system, discovered in 1946. This system is based on KEL antigens, encoded by the KEL gene. After the ABO and RH systems, the KEL system ranks 3rd in importance due to its potential to cause significant transfusion reactions and erythroblastosis fetalis. Of the 25 known KEL antigens, 6 KK, KPA, KPB, JSA, and JSB are clinically significant. So while the ABO and RH systems might be the most well-known, the MNSs, P-Lutheran, and KEL systems are equally fascinating, each with their unique antigens and roles. Understanding these various systems not only deepens our knowledge of human biology, but also impacts clinical practices, particularly in blood transfusion and pregnancy. These systems, although less known, play significant roles in our health. So, let's recap what we've learned about blood groups. We delved into various blood group systems, each remarkable in its own way. The ABO system, the oldest and most significant, divides blood into 4 types: A, B, AB, and O. Each type is distinguished by unique antigens, influencing how our bodies react to foreign substances. Next, we examined the RH system, another clinically significant group, splitting blood into RH positive and RA negative. Just like the ABO system, the RH factor plays a critical role in blood transfusions and pregnancy. We also discussed the MNSs system. Discovered in the late 20s, it's based on 5 key antigens, though it recognizes up to 50. While the system's clinical significance is generally low, it remains a fascinating testament to our body's complexity. Then, we ventured into the P blood group system, which classifies blood based on P-type carbohydrate antigens. Despite only having 5 antigens, 3 of them are important and can cause transfusion reactions and erythroblastosis fetalis. We also explored the Lutheran blood group system, with its 19 antigens that reside in our red blood cells' membranes. This system, though seldom linked to transfusion reactions or erythroblastosis fetalis, adds another layer to our understanding of human blood. Finally, we looked into the KEL blood group system. After the ABO and RH systems, KEL is the 3rd most important, with potential to cause significant transfusion reactions and erythroblastosis fetalis. Of the 25 known KEL antigens, 6 are considered clinically significant. These systems, each with their unique antigens and antibodies, demonstrate the intricate nature of our blood. It's not just a matter of being ABAB or O, positive or negative. It's about understanding the complex biology that keeps us alive, the intricate dance of antigens and antibodies that happens every time we face an infection, undergo a transfusion, or even during pregnancy. Remember, understanding blood groups is not just about knowing your type, it's about appreciating the complex biology that keeps us alive. Until next time, keep learning and stay curious.